Every year since I've joined OSU, I attend a 5K run called Purple Strike that raises funds for pancreatic cancer. It is an amazing event where people from different backgrounds come together to not only raise funds, but raise awareness for this disease and celebrate all of the survivors. It is an event filled with so many emotions. You get to hear the testimony of the pancreatic cancer patients, the survivors, their families and friends cheering on them. It definitely increases the sense of responsibility and drive to be the best researcher I can be. A little bit before the 5K run starts, they gather all of the survivors in this podium and one by one start saying how many weeks, months, or years they've been without pancreatic cancer. And this is incredible to hear, but the fact that all of them fit in this tiny little podium is heartbreaking. And this is because there's only a handful of survivors. Pancreatic cancer is a deadly disease. Once you get a pancreatic cancer diagnosis, you may only be looking at a couple months before succumbing to this disease. The main reasons for this is because pancreatic cancer lacks early detection biomarkers to be able to diagnose this disease early. In addition, it has a very high incidence of metastasis that worsens tumor outcomes and lacks any successful therapies. In the Cruz Monserrate laboratory at OSU, I researched a very common and deadly type of pancreatic cancer called pancreatic ductal adenocarcinoma. In this lab, we've also identified a molecule called Lipocalin 2, or LCN2, that is increased in the blood of pancreatic cancer patients compared to healthy individuals. In addition, this molecule increases and promotes inflammation in the tumor, and we know that deleting this molecule in preclinical models delays tumor growth. We also know that Lipocalin 2 is able to bind iron in complex with a siderophore, that are small molecules that come from bacteria. And because it's able to bind iron, it's also able to transport it to other tissues in the body, including tumor. And this is important because iron does so many things for our body. It's involved in metabolism, cell regulation, DNA, hemoglobin, and we know that in cancer, in order to sustain tumor growth, iron requirements are increased. And I've just talked to you about how iron can also interact with molecules like Lipocalin 2, and I will show you how by modulating genes like NDRG1, which is an anti-metastatic gene, it can also be involved in metastasis. The first thing I did to study this interaction was to measure LCN2 levels in 2 pancreatic cancer cell lines that either have or lack LCN2. And you can see here in the first panel that by adding iron, LCN2 levels are increased in the cell line that has LCN2 expression. In addition, we measure NDRG1, which is this anti-metastatic gene, and you can see that just by comparing the 2 black bars, NDRG1 is increased in the cells that don't have LCN2. And as we add iron, NDRG1 levels start decreasing. This result suggests a potential role for iron chelators in pancreatic cancer treatment. We also measured the same marker, NDRG1, this anti-metastatic gene, in 2 human pancreatic cancer cell lines, BX species 3 and PANC1. And you can see that as we add iron, the levels of NDRG1 are decreased. But when we reduce iron with an iron chelator, NDRG1 levels are elevated significantly, which is beneficial. We also corroborated this by measuring epithelial to mesenchymal transition markers, which are markers of migration. And we measured SEB1, SNE1, and TWIST, and you can see that when we add iron, these markers are elevated, while the epithelial marker, CDH1, is reduced. And this means that the cells will have an easier time migrating, detaching from the tumor, and arriving to other tissues to metastasize. But look at what happens when we reduce iron levels with an iron chelator. The markers SEB1, SNE1, and TWIST, which are epithelial to mesenchymal transition markers, are decreased, meaning that the cells will have a harder time migrating and metastasizing. So how can we take advantage of what we know about iron, how it interacts with LCN2, siderophores, NDRG1, and even bacteria that are the ones providing these siderophores to improve the outcomes of pancreatic cancer patients? Well, I am proposing to study this in preclinical models where I will determine how increasing iron levels in the tumor will modulate the interactions with pro-inflammatory molecules like LCN2 and migratory processes in order to decrease inflammation and metastasis. And this might improve patient outcomes and other treatment strategies available for patients. In addition, another aspect of my project focuses on determining whether these siderophores that come from bacteria are able to be used as early detection biomarkers to this disease to start treating the patients at an early stage. So going back to my mission and the mission of my laboratory, In the last Purple Strides, there were only a handful of survivors in that podium, but I see a day in the near future where everyone that gets diagnosed with pancreatic cancer will have many successful therapies to choose from and will be able to survive this disease and lead a healthy life. And in future Purple Strides, I will not see only a tiny podium filled with survivors, but the entire field crowded with survivors cheering on each other.